Biological Society meeting. Thanks for coming out tonight, and thanks for wearing mushroom gear. What a great outfit you've got. Tonight's speaker is Brian Vagg. He's a soil biologist whose company is called Sprouting Soil Corporation, and he's from Newport. And both Molly and I met Brian and Shelby at the Yachats Mushroom Show, and then subsequently another month later at the Depot Bay one-day mushroom event. And I saw his presentation of both of those mushroom events and thought we needed to see it here. So please give Brian Vagg welcome. Eugene CMS, welcome.
So as Chris was saying, my name is Brian Vagg. I am a soil biology consultant. Some of you may be familiar with Dr. Elaine Ingham. I trained with her. And so really part of what my wife and we created a company called Sprouting Soil. And that's my wife, Shelby. Hi, Shelby. We created a company called Sprouting Soil. And really what the goal of that company was to help predominantly agriculture, but really any kind of growing system convert from traditional agrochemical kind of management of crops to farming or managing crops with biology as a focus. And so really having an understanding of what's happening in the soil, all the relationships between the plants and the organisms was really, really important for our clients and our farmers to be able to understand so they can actually manage their crops appropriately. And with that, Shelby and I also were recently moved to Newport, and we joined the Lincoln County Mycology Society. And so, yep, we were on our way to becoming mushroom heads. It's funny. I look at fungus all the time. Mushroom nerds, I like it. I look at fungus all the time under my microscope, but I still am learning a lot about identifying mushrooms and doing the foraying and all that kind of good stuff. Okay, so let's get right into this. Oh, and also the way that the presentation is going to go today is we're going to go through presentation. And you can ask questions if you want to during the presentation. I'll also ask questions at the end of the presentation. And then what we'll do is we'll transfer over to the microscope. And I've got some samples of different soils. And we'll go take a look at what's living in there. And also in one of the soils, what's not living in there. It's a really degraded soil. Okay, this slide, many of you may have already seen this slide. It's actually a slide that Dr. Ingham created. It's published in the USDA site. And there's a concept called the soil food web. And really what the soil food web is trying to describe is the interaction of plants, which are going to be this kind of first level of organisms here, working with all of the organisms in the soil. And we have different kind of what we call trophic levels of the organisms, but we have two really, really important organisms that do a lot of heavy lifting in the soil, bacteria and fungi. And all of us are here because of fungi, but bacteria is another organism that's very, very important. And really what the role of bacteria and fungi are, they are basically the miners of the soil. I mean, I'm using mining in air quotes. They have the ability to create enzymes that will etch and mine away nutrients from sand, silts, clays, bedrock, pebbles, organic matter, all the components that make up the soil. And then from bacteria and fungi, we have other organisms that will then eat the bacteria and fungi. Protozoa, single-celled like amoebas and flagellates and ciliates. We have nematodes. They'll eat bacteria and fungi as well. But then we have the things that eat the bacteria and fungi. This is all part of this relationship we call the soil food web. I'm going to come back to this slide. This slide, there's a lot of stuff in here, but once we start talking about the mechanisms of how the organisms actually interact with plants, this will make a lot more sense to you in just a minute. Okay, so let's talk about bacteria. And when we do the microscope, I'll point that out. We have many different types of bacteria. A lot of times, morphologically, we talk about their shape. So there's cocci, which are going to be round bacteria. There are bacillus, which are going to be rod-shaped bacteria. There's actinobacteria, which is a filamentous bacteria. It creates almost like a fungal hyphae, but it has all these little pearls of actinobacteria that live inside the filament. We have chained. They'll come together in chains. I also, this is not a technical term, but I call them thrusters. They're very modal organisms, bacteria. They'll move in very distinct ways inside of the soil. And this key thing here, the ability to mine nutrients, this is an important topic that we're really going to come back to quite often, is that ability of bacteria to create enzymes that can mine the nutrients or etch away those nutrients from the soil. And then also there are some specialized types of bacteria that help you fix So have you ever heard of a nitrogen fixing plant? Like clover, peas, beans, your legumes, and so forth. So there's a type of bacteria called a rhizobium bacteria. And what the plant does is says, you know what, I'm going to allow that bacteria into my root zone, and I'm going to isolate it, and then I'm going to feed it a whole bunch of very high energy products. And what's going to happen is that bacteria is just going to multiply and multiply and multiply. And then pretty soon that rhizobium bacteria has the ability to take atmospheric nitrogen, which is a really, really strong chemical bond, and break atmospheric nitrogen apart and provide nitrogen back to the plant. Nitrogen is a key mineral for a lot of plants, but it's hard to come by. Rhizobium bacteria is actually a pretty cool bacteria for that. All right, let's talk about fungi. So fungi, there's so many different types of fungi. There's over 100,000 different species of fungi that are on this planet. And one of the ways that we look at fungi is they create these fungal hyphae. And in fact, you can see it right here. I think I have a laser, right? Right there, perfect. You can kind of see this filamentous, and we're going to see this when we actually look in the microscope. That's a fungal hyphae. And really what the fungal hyphae is just a channel that the fungus creates to protect itself and also to create a highway to be able to move water and nutrients, and also the growing tip. And that's that fungal hyphae. Fungus can be beneficial to plants. In fact, there's a lot of very good beneficial fungi out there. There's also a number of disease-causing fungi. I'm sure many of you who've grown plants have come across all different types of disease-causing fungi. And we'll talk about kind of this role here. You know, there are some fungi, in fact, we were talking about this in an earlier discussion, that will actually parasitize plants when plants are under stress, but the plant's not under stress, it will perform a different role. It'll be more of a beneficial fungi to that plant. And we also have some species that actually create a symbiotic relationship. You remember that rhizobium bacteria I talked about, how the plant will allow it to, and we call it infect the root? There's mycorrhizae fungi that will actually do that similar type of thing. There's mycorrhizae fungi that will actually infect inside the root. And there's ectomycorrhizae fungi that actually will surround the root in what we call a harding net. It's a really dense net of fungi just right around the root zone itself. And again, one of the key concepts here is the ability to mine nutrients from mineral and organic sources. So bacteria and fungi are two cornerstone organisms for this relationship with plants. Okay, well, we also talked about the predators, things that eat bacteria and fungi. Well, protozoa actually eat bacteria, and they're single-celled organisms, and we have quite a few different types. There are flagellates, and we're going to see them for sure when we look through here. And I like to refer to them as almost like they bumble through the soil. You'll see them, they move, they kind of bumble all throughout the soil. We have amoeba, which kind of ooze throughout the soil. And hopefully we'll find a naked amoeba because they just kind of ooze all over the place. And then we have ciliates. I like to refer to these as kind of the race car drivers of the soil. When you're looking at a microscope, you'll see a ciliate just zoom right by and then zoom by again. But if they find a lot of bacteria that they like to eat, they'll stop in one spot and then they'll just graze. And you can watch them actually just pulling all kinds of bacteria into their bodies. Now, this is also a key function of protozoa, consuming the bacteria. All right, so let's just go back to this concept we started to talk about nutrient cycling. Bacteria have enzymes. They break down nutrients. They consume the nutrient. It's now in their body. Is that available to a plant? No. So right now, that copper, that molybdenum, that whatever nutrient that bacteria consumed is stuck in the body of that bacteria. Well, we need something to eat the bacteria to unlock the nutrients. And in fact, when a protozoa consumes a bacteria, that bacteria is usually higher in the nutrient value than what the protozoa needs. So it has to excrete the excess in the form of a waste. We refer to this as the poop loop. The idea here is that that protozoa is going to eat that bacteria, now has excess nutrients, and it's now going to put it out in a plant-soluble form. And this is how the basis of nutrient cycling actually works. All right, let's talk about nematodes. And we're going to see nematodes in here as well. They look like worms. In fact, you can see one right here. That's the nematode. And there's all different types of nematodes. In fact, some of you, if you're gardeners or farmers, nematodes usually have a really, really bad name in agriculture. It's because of these guys right here, root-feeding nematodes. And in fact, if you ever pulled up a plant root and it's just got all these lesions and big old knots and the plant's not doing very well, more than likely you have root feeding nematodes that are just attacking that plant. Now, unfortunately, they give nematodes a bad name. There are lots of other nematodes. Bacterial nematodes or bacterial feeding nematodes, what do they eat? They eat bacteria. Fungal feeding nematodes, what do you think they eat? They eat fungus. And then we have predatory nematodes. Well, they eat other nematodes and they also eat protozoa. And so again, it's the whole idea here is that when a nematode eats a bacteria, too much nutrients, so it has to excrete out that waste. And now we have that poop loop going. Fungal feeding nematodes. These are interesting. They actually have a spear right in their mouth part, and they'll go up to a fungus and they'll puncture the fungal hyphae, and then they'll suck out the growing tip of the fungus into their body. And that's a fungal feeding nematode. And people may look at this and say, well, but I don't want to kill my fungus. It's all about balance in this case. Just like we don't want to have the bacterial feeding nematode eat all the bacteria, the fungal feeding nematodes won't eat all the fungus. If you have an ecosystem imbalance, you have enough fungus to be able to feed those nematodes. And the populations will adjust themselves based upon the quantity of fungus that's in the soil. And then again, predatory nematodes are fantastic because again, they eat other nematodes. And if a predator nematode eats a fungal feeding nematode, that's the next step in that chain. So we're going to have things that are going to eat nematodes, microarthropods. And then we'll have things that will eat the microarthropods, arthropods. And it's all of this predation that happens in the soil that's unlocking that nutrient cycling. Okay. And then again, we talked about arthropods and animals. So there are tons of different types of microarthropods and arthropods, and we have worms, we have all kinds of animals that will burrow into the soil. And so all of that interaction that's happening is impacting that root zone. We want to have a very healthy ecosystem with very diverse sets of organisms. Okay. So now let's really kind of dive down into how this nutrient cycling works. So I talked about that bacteria and fungi can make enzymes that will mine the nutrients in the soil. Making those enzymes is a very energy intensive process. Bacteria and fungi cannot make their own energy. They're energy devoid. So they have to rely upon something in the soil that's going to give them the energy to make those nutrients or those enzymes. Plants are power factories. They take sunlight, carbon dioxide, and water and make extraordinarily energy-rich components. Carbohydrates, amino acids, and proteins. All these things are very, very high energy. So the plant can produce energy. Bacteria and fungi cannot. So here's the trade-off. That tree right there will take about 40%, maybe even up to 60% of all the energy it creates, and it will push it outside of its body as a food source to feed microbiology. We call this food source an exudate. And all an exudate is just a fancy word for carbohydrates, amino acids, and proteins in whatever form. And think of it like a baker. You can take flour, a fat, and a moisture, and you can make a cookie or a cake or whatever. The plants have the same ability. In fact, that plant right there has about 64,000 combinations of exudates it can make. And what it's doing is saying, you know what? I want to make an exudate that's going to feed the copper-loving microorganisms because I need copper. I want to go into a flowering stage and I need copper. So that plant is going to create exudates that are going to stimulate the copper-loving microorganisms, the bacteria and the fungi in the soil. When it pushes out those exudates, those organisms explode in population. They start mining lots of copper. Along come the predators, the protozoa and the nematodes, and they start eating those copper-loving microorganisms. And now all of a sudden there's a lot of solubilized copper that's in the root zone that the plant can now pull up into its system. And it does that for molybdenum, for iron, for all the nutrients that it needs. It will change the type of exudate that's going to push out in the soil to stimulate those types of organisms. So that's the trade-off. Plants have energy in excess. Bacteria and fungi have the ability to mine nutrients. It's this trade-off, the symbiotic relationship that occurs. And a lot of people kind of get stuck on, though, that, Well, okay, I just need to have bacteria and fungi. But again, remember, a bacteria or fungi, once it gets that copper, or whatever nutrient it wants, it's stuck in the body of that organism. We now need also the protozoa, the nematodes, the microarthropods, and on and on and on. And another aspect about this is it's not just, one type of organism. We can't have just, 10 types of bacteria and 10 types of fungi. That plant's expecting there to be thousands of species of bacteria and thousands of species of fungi in the root zone, each one of them fulfilling kind of a different role of what kind of nutrient that it's going to actually mine from the soil. All right. So one of the key things I want to point out here, plants drive the biology. They're the ones that when they know what they need, they're going to push out those exudates, and they're hoping that some microorganisms are home to go mine those nutrients for them. And this is, in the work that Shelby and I do, this is a common thing we run into, is that the plants are trying to drive biology, but there's no biology in the soil. The management practices that we've used have basically denuded all those really good organisms from the soil. So the plants are pushing out exudates, nobody's home, and they're not getting the return. The plant is now relying upon us to fertilize it, to feed it, and so forth. Okay, so just as a little bit of a review here, but we call this thing nutrient cycling. And really the cycle just goes around and around. Plants are going to release exudates to feed specific types of microbiology. Bacteria and fungi are going to mine the soil for and organic matter for those nutrients. The predation of the bacteria and fungi is going to release those nutrients into the soil, and then we go again. And all throughout that life cycle of that plant, it's changing all those different types of exudates that's actually pushing out to that soil. All right, so let's also talk about a few other things about plants. A very important thing for us is around root depth and compaction. So, a lot of people, and I've talked to a number of corn farmers, we'll talk about how far do your roots go down? And they're like, oh, my roots go down 18 inches. They go down 12 inches. And then they stop. Well, what's happening is they've hit a compaction layer that won't allow their roots to actually go down. If they didn't have that compaction layer, those corn roots might go down 10 feet, 15 feet. This is a picture of prairie grass. And some of these prairie grasses are going down 30 foot in the soil. That plant may only be 16 inches tall, but it may have a root zone that goes down 30 feet into the soil. And a lot of people also think that, well, only the microbiology is going to sit at the very surface of the soil. That's not true. That plant, anywhere that root zone is going to be, is going to drive different sets of biology to be around that root zone, as long as there's biology there. So one of the things that we constantly deal with, especially in agriculture, is people plow. And you get what we call a plow pan, which is a hard layer of compaction. It's usually wherever the plow ends. So we'll find roots will go down and then they'll go sideways. And you're really limiting, if all your roots just existed right at the very top part of that soil, look at all that access to nutrients that you're not getting access to. So compaction is another major, major component that we have. Now, here's the other parts of good news, though, that the biology also deals with compaction. All right, so a common thing that we run into in a lot of agricultural situations is that we're going to run into compaction because the magnesium-calcium ratio is off. So clay particles, think of a clay particle almost like a pancake. When the calcium-magnesium ratio, and this is usually how much calcium-free ions of calcium and magnesium are in the soil, if the ratio is not correct, the clay plates are going to stack upon each other, just like this. But if we have the ratio set correctly, then they'll repel each other. And now we've opened up the soil. So have you ever heard of gypsum being put on a field, or lime? Farmers will call it sweetening the soil. Well, they're trying to address the calcium-magnesium ratio, right? And they'll do it for a period of time. But what happens when they put the gypsum down, rain's going to occur, irrigation is going to occur. What's going to happen to those nutrients? They're going to wash out of that system. And pretty soon you'll go from nice fluffy soil back to really hard pan, really compacted soil. Well, every time I had to keep on putting gypsum and lime, it's a cost, but I'm also adding a tremendous amount of salt to my soil. I'm killing biology when I do that. All right, so the thing that's near and dear to us is fungus. Fungus are great regulators of the calcium-magnesium ratio. If you have a good population of fungus in the soil, you're going to set that calcium-magnesium ratio to about the right level to what we call flocculate the soil. make those clay platelets stand and open up the soil profile. So biology also not only just does nutrient cycling, but it also creates this structure inside the soil. So one we talked about was calcium, magnesium. Another thing that the biology will do is it will aggregate the soil. And we're going to see that. I have an agricultural soil. And in fact, I'll just show you a little bit here. And you can see this soil is very pale in color. It has no structure. If I was to put this in water, it would just dissolve right away. Here is a forest floor. You can see the color is different. There's a lot of structure that's actually the soil has been made because the bacteria and fungi, not only do they cycle nutrients, but they also help aggregate the soil. The bacteria will create these glues that will start combining sand, silts, and clays and making little homes for the bacteria. Fungi go through and start wrapping all of those aggregates together into make what we call macroaggregates, bigger aggregation. If you've ever pulled a plant out of the ground and you see all those kind of like clods that stick, but you can take your hand and kind of crush them apart, it's because the soil's been aggregated around that root zone. So compaction is a major problem, but biology can also help us solve those compaction issues. All right, let's also talk about plant succession. So we know that bacteria and fungi, again, are primary for us to doing the nutrient cycling, but they also soils will tend to drive more bacterially dominant or more fungally dominant based upon where we're at in plant succession. So if we look here, very highly disturbed or, very what we call primary succession has happened, we're going to be almost all bacteria in that soil. As we move along into more like grasses and crops, typical annual crops, this can be more of a balance of bacteria and fungi that can be in that soil. When we start moving all along this chain to like an old growth forest, it's going to be predominantly fungi. In fact, when we start looking at the forest floor soils, you're going to see a lot of fungi. When you look at the agricultural soil that I have, it's going to be almost all bacteria. And so it's really important for us to understand, where do we want the biology to be set? If I'm going to grow, let's say, corn somewhere right around in here, I definitely don't want it to be massively fungal. I also don't want it to be all the way bacterial. I want to have a mix of bacteria and fungi in there. And what we do is we use our microscope to be able to assess that. Where do we have the balance of bacteria and fungi in that soil? And if it's not set for the crop that we want, then we're going to try to implement and put biology into the soil to try to get it to that successional level that we want it to be at. Okay. So there is a fungus among us. For many of you, you're probably already getting this almost like a refresher course, but we're just going to talk a little bit about fungi in particular. So fungal facts: fungi are eukaryotic organisms. There's approximately about 144,000 known species of fungi in the fungi kingdom. And I'm not a taxonomist, but I have a lot of respect for people who do fungal taxonomy because it is a crazy branch. There's always tremendous amounts of changes and arguments that are going on in this fungal community, for sure. And there's also a lot of many fungus-like species. So mycetes, we're going to talk a little bit about mycetes. A lot of them kind of end up being plant or disease-causing organisms that will impact plants. They're not actually a fungus. And this is actually kind of a newer thing. For a long time, people thought that mycetes were kind of in the fungal kingdom, but they're not. They're their own. Another fun fact about fungi: they're the most widely distributed organism found on Earth. Fungus almost lives everywhere. And then again, we talked about some fungi can form symbiotic relationships, some can be parasitic, and some can switch their role depending upon the health of the plant. You got to think about Mother Nature sometimes too. Having disease-causing fungi is not a bad thing from a nature standpoint. If you have a plant that's not genetically well, or it's not really suited for growing where it's going to be growing, we want organisms to take that out of the gene pool. That's the role of a lot of these disease-causing organisms. The unfortunate thing for us is that we're artificially causing a lot of stresses on our plants, which cause these disease-causing outbreaks to actually occur. Also, a little bit about the life cycle of fungus. You're going to see when we go through in the microscope, we're going to see spores, we're going to see fungal hyphae, and that's all going to be things that you and your naked eye will never be able to see. In fact, most of us, when we're looking at fungus, we're looking at the mushrooms. And this is part of the sexual reproduction aspect of it. And it's just a small part of the life cycle of actually of a fungus. A lot of the fungus is going to be happening underground, unbeknownst to us. We cannot actually see it. Some other kind of general characteristics of fungi, and this will help us when we start looking through the microscope. So, and I want to just put a caveat in here. When I say beneficial fungus, mycorrhizae fungi, and disease-causing fungi, these are just some general guidelines. There are always exceptions to these rules. There are some disease-causing fungi that will look a lot like beneficial fungi. There are a number of fungi that look like disease-causing fungi, but they're perfectly beneficial to the plant. So I just want to point that out to everybody. A couple things though, when we're looking for fungi, and in fact, when I'm looking at, say, some beneficial saprophytic fungi, and is everybody familiar with the term saprophytic? Decomposers, right? They're going to decompose things. A lot of those are going to have uniform diameter hyphae. So we saw that one picture where the fungal hyphae was there. The fungal hyphae should be uniform. It should be 5 micrometers at one end, 5 micrometers at the other end. You know, there should be no difference as far as uniformity when we're looking at more the good beneficial fungi. Generally, they're going to be about 3 micrometers in width or larger. There are some, in fact, we'll see in some of the forest floor, there will be 10 micrometers in width. Huge fungal hyphae. Also, a lot of the beneficials will have some kind of color to them. Tans, browns, purples, all different shades of tans and browns and so forth. Now, are there saprophytic fungi that are clear in color? Sure. And then also, another thing about fungi in this particular class is we're looking at uniform distance of septi, and all septi are cross walls. So as they lay down that pipe of the fungal hyphae, a lot of fungi will put down cross walls. It just helps stabilize the fungal hyphae itself. And one of the common characteristics of the beneficial saprophytic fungi is that it's uniform spacing of those septi that are in there. And then we also have, dormant stages. There's asexual spores, sexual spores. There's as far as the reproduction, it's all across the board. Now, mycorrhizae fungi, we talked about that one as well. That's the fungi, the endo or the ecto mycorrhizae fungi that will actually make a direct relationship with a plant. They're typically 3 micrometers in width as well. And again, this is the width of the fungal hyphae. Many different colors. From light tans to dark browns to purples to all kinds of colors. They may or may not have septi, but if they do, they're usually going to have some in uniform in presence. But not all of the mycorrhizae fungi will actually create septi. And then when we look at the disease-causing fungi, these usually kind of look lumpy. They don't have uniformity to them. If they have cross walls, they're usually just kind of all over the place. They're not usually in a uniform kind of spacing. And a lot of the disease-causing fungi are actually in that 1.5 to 2.5 width. And we'll talk about that when we actually look in the slide. And they're almost always clear and colorless. So when we start looking in the scope and I start looking at soils and I see a predominant of this type of fungi and not this type of fungi, I can start making some assumptions about the quality of that soil. Why do we have all these disease-causing fungi in that soil when we really want to see more of this kind of fungi in the soil? All right. And again, we talked about a little bit about the fungal hyphae. So when the fungal hyphae, it starts right when the spore germinates. In fact, it's really cool. And hopefully we'll see one in here. We'll see a spore that's germinated and we'll see the growing tip of the fungi coming out of the spore. And from there on out, it's going to go ahead and create that fungal hyphae. And then there's a cytoplasm and it's concentrated in the tip. And I want you to think of like this. The cytoplasm is growing. It's laying down this really nice carbon structure, the fungal hyphae behind it. And it may go all the way down this branch and then realize, you know what? The food sources aren't that great there. What it will do is it will stop growing. Cytoplasm will move back through the fungal hyphae and say, you know what? This probably is a good spot. I'll create a branch. And then it will start growing off this way. And you'll see commonly when we look at fungal hyphae, all this branching that's occurring inside of the soil. And the waste products of that, it's really rich in carbon. So a lot of us are concerned about carbon in the atmosphere. Fungi are great sources to sequester carbon. This fungal hyphae is primarily made out of carbon. And if you look at forest floors, tons of fungi, we're sequestering a lot of carbon right back down into that soil. And let's see. Yeah, that's about it with that one. And then also just talk about fungal spores. We're going to see fungal spores. The one thing I wanted to talk about though is a lot of us see spores. We'll do spore prints with mushrooms, but there's a tremendous amount of spore development that can happen underground in the soil and not in a fruiting body like a mushroom. Even a truffle is a fruiting body. But even, and we're talking microscopically, you can find these little tiny sacs that will contain all these spores. In fact, some of them are really cool, like the zoospores. When they explode in the soil, in fact, sometimes you see them, they're modal. They'll just go crazy. They just start swimming everywhere in the soil. If we see one, we'll be lucky. But if we do, that'll be fantastic to see. Okay. So I know I went through a lot of topics and I wanted to see what kind of questions you guys have. Don't feel shy. It could be 20 micrometers in size. Yeah. Oh, sorry. Yeah. So the question was, what size can that spore sac be inside the soil? A huge variety in size. Some of the sacs can be 20 micrometers, 40 micrometers, 60 micrometers in size, contain tens of spores, hundreds of spores. Each variety of fungi that are going to do usually asexual reproduction, including those spore sacs, are going to be different. Questions? Yeah. Great question. So a fungal hyphae, and let me just actually go back to this real quick. So that's a fungal hyphae, right? So that's a single, carbon pipe. Mycelium is a combination of a bunch of fungal hyphae together. And in fact, when you go out into the forest floor and you start digging around in all that wood duff, and all of a sudden you pull out a wood chip, and then you have like 6 wood chips hanging from this white stringy stuff that's in there. That's mycelium. And it's really cool to see under the microscope. You just see all this interweaving of fungal hyphae on top of each other. Good question. Yeah. Get incorporated back in the soil. And that can happen many ways. Animals that burrow, that dig, disturbances that happen will put those spores. They'll lay on the surface of the soil. Rain events will help filter them down into farther down to the soil profile. Yeah, so many different ways that spores can actually travel. Again, wind-blown spores, they'll travel wherever they fall. A lot of times rain or animals or so forth are going to incorporate them into the soil profile. That's a great question. I don't know. I don't know if certain species have a better ability to be able to put, especially like the wind-blown ones, the ones that are above ground and going down in. Are some spores better at getting farther deeper into the soil profile? I don't know. That's a great question to ask. But a lot of times it has to have some mechanism to be able to help it get down into that soil profile. And probably rain's going to be one of the bigger events, just being able to wash that down through that question. So the question was, how fast can this nutrient cycling occur? So, and let me just expand upon your question. Tell me if I got this right. So when a plant says, I need copper at this time frame, and it starts pushing out copper X, or the exudates are going to feed the copper-loving organisms, how long until copper comes back to that plant, to that tree? Was that the base of the question? Okay. It's not seconds, but it's within hours it can start that process. And one of the nice things about this is bacteria can reproduce like crazy fast, right? So you start feeding bacteria food, they'll start multiplying, multiplying, multiplying. The more bacteria that you have that has energy to be able to make enzymes will start that process right away. But we had to have the protozoa in there, things eating the bacteria and so forth. So that can happen in a very, very fast time frame. And the plant, it's kind of, receiving. I got copper coming in, copper coming in. I put copper out for my flowering or whatever it's going to use the copper for. And it's not like, yeah, you know what? I now need molybdenum or iron. And the reality of the story is the plant's going to need copper, molybdenum, and all these things in different quantities all throughout the growing cycle. And in fact, what's really kind of interesting is one part of the rhizosphere, the plant root, might be putting out a lot of exudates for copper-loving organisms, while another one's putting out for molybdenum from the same plant. So all over the rhizosphere, that's the root zone, it's pushing out all different types of exudates to try to get the nutrients that it needs. But it's in control again. It's the one that decides what types of exudates it's pushing out to try to stimulate those types of organisms. So the question was, a difference between like agricultural soils versus like timberland soils. The second question was, is there a way to revitalize those forest lands that are more like the industrial monocropped kind of timberlands, right? Can we actually repair those? By all means, we can. The challenge, when I go look at forest land, like an old growth forest that's been fairly undisturbed, when I look at the fungi in there, massive amounts of species diversity. You can just tell by the different colors and the size and the spores and so forth. When I go look at timberland, it's much reduced in the variety of fungi that's there. One, again, you have a single species of tree that's kind of figuring out what fungi it likes, but then also those timberlands are in high disturbance areas. They'll cut them down, they'll slash them, sometimes they'll burn the piles. The organic matter is getting removed from the soil. And so we run into more compaction problems and things like that. Can you restore those lands? For sure you can. And one of the ways I really didn't mention in this talk, but one of our vehicles of how we actually repair degraded land is to add biology back into that system. Now, you probably have seen, you can go online, you've probably gone to nurseries, and you can find products that say it has 10 species of this type of bacteria and 5 species of fungi. Well, when we're really talking about healthy ecosystems, we're talking about tens of thousands of bacteria. We're talking about thousands of species of fungi to make a healthy ecosystem. So unfortunately, those products only get you so far. And plus, those products don't have the protozoa and the nematodes and all those other things that help with nutrient cycling. An amazing product that we can make is compost. And we can make compost in many different forms. Vermicompost, which is compost with worms. Thermophilic compost, which means making compost where we're managing time, temperature, and moisture. And we are going to make compost to try to drive the beneficial fungi, beneficial bacteria, the protozoa and the nematodes and so forth. I'm going to show you, this is a compost that I have right here. That came out of our vermicompost. When we look at this, you're going to see tons of flagellates and amoeba of all different types, rods and coccy's of all different shapes and sizes of bacteria, and many different types of fungi in here as well. So I'm very mindful when I'm making this compost to make it in a way that's going to foster the beneficial soil microbes. And I'll give you another hint or a trick here. The microbes we're talking about, this fungi, the bacteria that we looked at earlier, the ones that really are going to interact with plants are going to be aerobic organisms. They need oxygenated environments. Unfortunately, a vast amount of the compost that's created or produced is made anaerobically, and it produces anaerobic organisms. Anaerobic bacteria is predominantly going to be in those composts. So I hope you don't walk away from this and say, oh, great, compost is our tool. Let's just go down to the local Walmart or wherever you're going to go to, buy compost, and then expect that you're going to get this kind of benefit from it. Unfortunately, that's just not the case. We have to change the way we make compost. And I'll tell you the dirty little secret about compost, predominantly that we can buy. It's a waste management problem. Composters do not make their money from selling compost. They make their money by receiving organic matter in. It's called tipping fees. That's where they make the vast majority of their money. Compost is a waste material. They just want to make it and get it off their lot so they can bring in more material to make compost, that tipping fees. What we're trying to do is trying to work with these large scale composters to change the way they make compost so that it's made in an aerobic fashion to foster the organisms that are going to benefit the plants. Making anaerobic compost is usually not that great for plants, and in some cases can kill or harm plants. I have bought commercial compost, put it into growing systems, and watched plants just wither or really struggle or get massively stressed. It's the wrong type of biology that we're trying to put in there. And in some cases, it's not even just the biology. It's gone so anaerobic that it's creating alcohols and other really nasty products that will really harm plant roots. Question? Compost? Sure. So, how do we make aerobic compost at home? Sandy, thank you so much. You're keeping me on track here. The question was, how do we make aerobic compost at home? Well, I'll tell you, and I encourage everybody, when we get to the spot where we're going to look at this or at the end of the session, come up and take a look at these products, smell and sniff, especially this. This is vermicompost. So I made this with worms. Worms are fantastic. Composting worms will do a great job of breaking down organic matter, and they really will foster a lot of really good biology. Now, do I still have to pay attention to my vermicompost? Can I make Bad vermicompost, you bet. I've seen horrible vermicompost. Too wet, too anaerobic. So there's a technique to it for sure. Also for home composters, there's a type of composting that we make called thermophilic composting. And it's a fancy word, but it means managing time, temperature, and moisture. And when we do this, I want to build a recipe. And have everybody heard making compost what browns are? Woody material. It's high carbon, low nitrogen. We're talking wood chips and straws. Things are going to be 100 to 1, 100 parts carbon, 1 part nitrogen, 200 to 1, 200 parts carbon, 1 part nitrogen. 60% of my recipe is probably going to be in browns, a lot of woody material. And that actually is what fungi love. You think about it, in forest floors, it's all that organic matter, all those browns are just decomposing away. 30% of my recipe should be in the form of greens. These are going to be grass clippings. When you go out and you prune your plants, all the green, things, leaves that are green that have fallen off the plants, or you've removed off the plants. The C to N ratio there is about 30 to 1, 30 parts carbon, 1 part nitrogen. So now we're a little bit more what we call a higher nitrogen product. The last 10% of our recipe should be around a high nitrogen. We call a high nitrogen product 10 to 1. This is going to be manure could be used, grains, cracked corn. A fantastic source that I used to like to make with my compost, beer mash. Brewers have a waste product that's fantastic for composters. Beer mash is around 12 to 1. Fantastic. So I go to my local brewery. They have to actually pay to get rid of their beer mash. You can take that from them and they actually use it for compost. So we have a recipe now, 10% high nitrogens, 30% greens, 60% woody. And let me just tell you that recipe are guidelines. If I'm composting cold, I add more greens and more high nitrogens. If I'm composting in hot weather, really hot, I'm going to add more woody material and reduce the amount of high nitrogen and greens. Okay. All right. So the recipe is the first part of this equation that we have. The next is saying we now need to mix all those components together and make sure they have enough water. So moisture is a key part of this. Organisms, all these organisms that are living in the soil, they need moisture. Moisture is key to them. So we want to run about 50% moisture in our piles. I'll give you a really easy trick. Use your hand, reach into a pile of whatever it is, in this case, compost. And if you reach in there and you squeeze it really, really, really hard, and you see a thread of moisture going between your fingers, that's 50% moisture. If you squeeze really, really hard and you get nothing out of there, but you open it up and it sticks together still, that's probably around 40% moisture. If you squeeze really, really hard and you start getting some drips, 60% moisture. Just barely starting to squeeze and it's getting drips, 70% moisture. You start picking it up and it's starting to drip, it's 80% moisture and so on. 50% of moisture is where we want to be at. You get too much moisture, there's not enough oxygen. You'll go anaerobic. If you don't have enough moisture, it's not enough for the organisms to grow in. The pile will shut down. It actually won't compost. It will just stay dry. Now, the last part of this is going to be time. So what we'll do is, and temperature, sorry, it's temperature and time. We will take a thermometer and we'll measure the temperatures. And we have some guidelines in here. We'll say when the temperature ranges between, let's say, 131 degrees Fahrenheit to 150, we want 3 days of that temperature in the core of that compost. After 3 days, we're going to turn that compost. If it gets to 150 to 160 degrees Fahrenheit, we're going to wait 2 days, and then we're going to turn the compost. If it's 160 degrees and above, we're going to wait 24 hours and turn the compost. If we hit 170 degrees, we're going to turn it right away. Because once we get to 180 degrees, we're going anaerobic. And also what will happen is alcohols start getting produced. Has anybody heard of composting fires? You add way too much high nitrogen material into a compost pile, it goes crazy, crazy hot. All these bacteria just grow like massively and all the oxygen get consumed. It's now gone anaerobic. Anaerobic organisms take over. They love to produce alcohols. Piles running 180 degrees, producing a lot of alcohol. 180 degrees is the combustion point of alcohol. Boom, pile will go up. Now, I don't want to scare anybody because you have to really kind of mess up your pile and really not get your recipe right, not be paying attention to get it to a spot where it's going to go to a fire. But still the same thing is if you're not paying attention to your pile, then it's going to go anaerobic and you're not going to make great compost. Now, with that being said, if I have my recipe right, I'm probably turning my pile between 3 and 5 times. After that, I've consumed the energy and it will now just go back down to ambient. It takes between 21 and 28 days to do the active composting. And you can start using that compost within about 2 months. Yep. Home composters. Yeah. I have a number of clients that are completely. Yeah. And in cold weather, like if I'm going to compost in winter time frame, I might be like 13% high nitrogen, 33% greens, and maybe 53% in the woody material. So I adjust my recipe to have more nitrogen to combat the coldness. Because again, temperature is an important factor. But if I'm composting in California down, it's 110 degrees outside, I do not want 10% high nitrogen, 30% greens, because that pile is going to get way too hot and it's going to be hard to manage. So I reduce the amount of the high nitrogen and green material. And I have a number of clients that are just homesteaders doing their own gardening kind of thing, and they make thermophilic compost like crazy. Once you get the process down, it's easy to do. It takes a little bit of time and you have to learn the process, but once you learn the process, it's pretty good. And that's why I think vermicompost is probably maybe superior for a lot of home people, because worms are pretty easy once you learn how to manage worms. Constantly just feeding and making sure that, one thing that I find a problem with vermicompost and home people doing vermicompost is they forget to add in carbon sources. You need to make sure you're adding in some wood chip or some straw or paper bags or something, some high carbon material to help offset. Because if you don't, it's going to be too much nitrogen. You're going to start to go anaerobic. It's just going to be kind of a mess. But with anything, you at home can make fantastic compost. And that is your inoculum that you're going to use to be able to go and put it into your plants. And I'll just finish one last thing about this. So you've made this wonderful compost, or you have access to really, really good compost. How do I get it to the root zone of that plant? In fact, let me just go back here. All right. So I have plants right here. How do I get that compost to this plant? Well, if I'm planting a plant or I'm doing something from seed, easy peasy. My starts, I can add in compost, not a problem. When I go plant them in the ground, I can add a little handful of compost, the physical form of compost, great. But what happens when I have a yard full of all these plants? I've already been growing there for a long time frame. Digging up the plant, trying to add compost into it, or even top dressing may not be very easy to do. What we can do is we can take that compost and we can make an extract. And we call this a compost extract. And simply all we're doing is we're taking compost, that has all this really good biology, and we put it into a mesh bag, and we're going to wash off the biology from the surfaces of the compost. It's now going to take all that bacteria, fungi, nematodes, microarthropods are going to get washed into the water, while the physical part of compost, all the organic matter, gets removed from the equation. Now I have a liquid that's full of all these organisms. All I have to do is then water that in to my plants. You can water can it. If I have particularly hard compacted areas, I will use something called a deep root zone wand. It's just a big pole that has tubes, or it has openings at the end, and I push it down into the soil, hooked to a pump, and I pump the extract down into the soil profile. So there's a lot of different ways you can actually impart biology into to the soil. There's one last thing I do want to talk about, though, too. Remember I talked about exudates being pushed out of the plant in the body? Most of you are probably thinking, well, the exudates are going to come out in this root zone. What's really crazy is that the plant will put exudates out on the surface of the leaves, the bark, the stems. Every surface of this plant is going to push out exudates. Plants are nothing but efficient. If it's pushing out exudates onto the leaves, onto the flowers, what is it expecting? It's expecting that biology is going to be there. It wants to feed the biology that's going to live on the surface of the plant. So let me tell you about disease. We use what we call compost teas. So remember that extract I talked about where we wash the biology off? You take an extract, you now add food, and you add air to the equation, and you exponentially grow biology into liquid. Now, I might brew it for 24 to 48 hours. When I'm brewing it, those organisms are creating biological glues. It's liquid, but essentially it's sticky. And what I want to have occur is, let's say I have a leaf right here, and I spray compost tea on the surface of the leaf. As that drop hits, all those biological glues are going to help adhere bacteria, fungi, nematodes, all kinds of stuff to the surface of the leaf. So this leaf, I now sprayed it. Everywhere on the surface of this leaf has been sprayed with this compost tea. Now you have a bunch of organisms living on the surface of this leaf. And the plant is also pushing exudates out to the surface of this leaf to keep on feeding that organisms. All right, powdery mildew spore comes floating through the air. And I'm sure most of you have dealt with powdery mildew in some form or way. It lands on the surface of this leaf that's now been covered with all this great microbiology. It has to compete with all those other organisms to get access to those exudates. And in fact, these aerobic organisms are going to outcompete that powdery mildew. Powdery mildew has no chance, can't actually attach to the plant. All right, let's take an agricultural scenario where I don't have any biology on the surface of that leaf. Powdery mildew goes floating through the air, lands on the surface of this leaf. There's no competition, tons of food. What happens to powdery mildew? Explodes. I mean, I've seen farms, powdery mildew, I see it one day, and it's like, okay, we've got to deal with that. And 3 days later, the fields are just covered with powdery mildew. It will really just run like wildfire through your plants. So we use compost teas as a foliar aspect to help prevent and protect plants from diseases, like fungal diseases. So biology can be a really, really useful tool for us to help us managing crops. In fact, that's essentially what we do is we say, let's find ways to remove the chemicals from the equation. Your pesticides, your herbicides, your insecticides, your fungicides, on and on and on. And we're going to replace them with biology. In the same aspect, we're also going to create a healthy root zone so the plants can start doing nutrient cycling for themselves. I have clients that actually don't use any chemicals at all anymore. They simply use compost and compost derivatives like extracts and teas, and they're managing their plants. So the question was, what's the best way to get a beneficial fungi back into your soil? The answer is, what beneficial fungi are we talking about? If we're talking about a saprophytic fungi, then what we're going to want to do is make sure that the compost that we're creating is chock full of these really good saprophytic fungi. And I'll tell you a trick that I like to use. When I'm making compost, that recipe I talked about, 10, 30, 60, part of that is it's a little bit of a misnomer. I'm usually going to go out to my natural ecosystem, and I'm going to take pinches of soil and the organic matter that exists on the surface of the soil from different locations, because I want to grab some species that are here, some species over here, some species over here. Really, in order for this system to really work, diversity is key. If I have a very low diversity of organisms and I put that, treat my plants, well, when the plant says I need copper, but there's really not that many copper living organisms around, it's going to be shy on copper. Well, now the tree's under stressors. And each one of those little micro stresses now open it up to diseases and viruses and those other types of things, right? So I want to make sure that the compost I'm creating is massively diverse. Go find the indigenous microorganisms that live in your environment. And you can find them all. Go to riparian areas, creek beds. You can go in the forest. You can go to, native grasslands and so forth, and collect a little bit of material from those locations. Put them into your compost when you're making your compost. And then you'll have those organisms. Now, I did say about fungi, it also depends. Mycorrhizae fungi, on the other hand, you're not going to go out there and develop them in your compost, because mycorrhizae fungi require that you have a plant in the system. In fact, a mycorrhizae spore is not going to germinate until it gets a signal from a plant. The plant's going to say, hey, root zone's over here. I'm open for business. The spore says chemically, hey, okay, this is good. Germinate. I'm now growing towards that chemical signature. Now I'm going to go ahead and make that connection with the plant. So there are some things we can do, though. When I make compost, I can now what we call make mother piles. A pile of compost that I'm going to now grow plants in and make sure that they've made mycorrhizae fungi connections. So I can go take a look at the root under my microscope, say, oh yeah, I see the endomycorrhizae fungi, or I see the ectomycorrhizae fungi. I want to take that material and add it into my compost. So now I have a bank of mycorrhizae spores that I can actually add in for whatever crop that I'm growing. So as an example, if I'm growing almonds, I'm going to grow almond trees in a mother pile of compost, check to make sure they made mycorrhizae connections. If they have, I'll use that mother pile as part of my component to go into the soil. And now I have a bank of mycorrhizae spores that I can actually use. The question really was, it's that I need to expound more on my discussion around industrial forestry in regards to how do you actually get the biology set appropriately for an industrial forest system, especially when we're talking about large swaths of land that are very difficult to get access to. And I'll tell you this, In agriculture, like fields and so forth, it's actually pretty easy to get biology to the root zone because I have planters, I have seeders, I have irrigation, I have all these different tools I can actually directly influence the plant. In forestry, I don't have those tools available to me. And it's not like I'm going to pay a team of people with backpack sprayers and try to go out there and inoculate. Aerial spraying is going to be a big thing that we use in forest ecosystem restoration. Drones now actually are a very key component. We have drones, agricultural drones, that will work well into the forestry system. Well, they'll just fly down, pull up the compost extract, fly out their pattern. When they're empty, they come back and they fill up, and they just keep on doing that. And you have an army of drones going out there and actually applying biology. It's actually being done, correct. Yeah, and this aerial aspect, when we're talking about large scale ecosystem restoration, is a key way to do it. There's also another way. When we're in forestry land and we have the ability for a person to directly interact with the forest land, is when we're planting those trees. So what we want to do is take those starts. We're going to soak them in compost extract. We're going to make sure that the root zone becomes well inoculated by the time it actually goes out into the field and gets planted. And so that's another way we do it. Hydroseeding is a way that we do it. I actually have a client that we're working with a tribe in Nevada. They're trying to reconstitute their sage and pinyon pine forests. Where we can't get our hydroseeding trucks to be able to put a slurry of compost out there, now we're using helicopters. Instead of spraying, it's a slurry that then gets spread out across the ground in these big buckets that hang underneath the actual helicopters themselves. So there are tools, but it's still pretty expensive to be able to do that. But it's very important. And what we can do, like a tool with the microscope, we can go in and assess a timberland and say, what is it missing? And predominantly, usually, when we have massive amounts of disturbance, it's going to be fungal. That's going to be a major component that's missing. And in fact, when we looked at the succession, I know our forest lands, we want to have mostly fungi. So I'm going to be doing all the work I can to make sure that the compost I'm using is high in diversity of fungus and also quantity of fungus and fungal spores. And so I'm making sure the products I'm using has those organisms in it before I actually go ahead and put it out there. I mean, really, for organisms, it can be more around moisture events than anything else. So if I was to say, go out and use those drones, when I want to go out and spray these into my forest lands, if I go out into the forest in, let's say, September or August, no rains, floor is dry, really, really dry. I take organisms that are going to be falling through the air, still in water, but boy, they're going to hit that really dry duff layer that's there. Most of them are not going to be able to go dormant. They're just going to die. So I want to time my events where I'm actually putting organisms out in there when I actually have moisture events that are going to occur. Moisture is more the critical component when we're talking about inoculating fields. And I have the same problem in agriculture. When we're doing agriculture, I'm usually trying to time my adding in or inoculating organisms when we're talking about water events. So that could be rain, irrigation, a lot of different ways, but water is the most important aspect. So the question is saying, hey, we had these major events that occurred, like the freezing rain, and a lot of disturbance or damage where we're having a lot of material that's now fallen onto the forest floor. How is that going to impact the ability for mushrooms or to develop, like the fruiting bodies of fungi? And there was a second part to that question, which was, and what steps are the forest going to take to be able to return to its balance? I really can't speak to as far as how that's going to impact the mushroom development. I really, truly don't know. And I'm sure there's some mycologists out there that would know that answer. Yeah, exactly. The fungus exactly is what's going to do the restoration work for us in that forest land. And in fact, what's happened now is you have a massive amount of organic matter that has just fallen onto the surface of the floor. What we're hoping is there's a lot of these really good beneficial organisms like saprophytic fungi. They're going to start, hey, let's decompose that material. Now, we'll talk about fresh material though. Pine needles that are green that have fallen on the forest floor. They take a little while to decompose. In fact, there's a waxy layer that's on the outside of all those needles. It takes a while for that waxy layer to be able to break down. And also, a lot of the conifers have these volatile compounds that are very antimicrobial. It's kind of its defense mechanism. So we have to wait for that material to start to off gas before we'll really get an influx of microbiology that's in there. And I'll use this. I'm going to take your question. I'm going to go one tangent off of there. And that is, a lot of people ask me this question about wood chips, using them in compost, using them in mulch, and so forth. And in fact, right now, wood chips are very prevalent. You can find wood chips all over the place, right? I typically, and I always say typically because there's sometimes, in fact, I'm doing it right now. I'm going against my rules. But typically, what I will do is I will bring wood chips onto my property and I will quarantine them for a period of time. The period of time depends upon the trees or the type of material that I brought in. If I have a bunch of cedar chips that have come in, they're going to take a lot longer in quarantine than if I get a bunch of alder chips. And really, the best tool that you guys have is your nose. If you can dig into a pile of wood chips and you go, oh yeah, that's cedar, or wow, that's eucalyptus. You know, the smell is strong. There's still a lot of those volatile compounds, those antimicrobial compounds existing. Don't use it yet. If you go in there and you smell, you're like, oh man, maybe cedar. I don't know. By that time frame, most of those volatile compounds have off-gassed. Now go ahead and use that material. Use it and you're making your compost, use it as a mulch and so forth. And same thing on the forest floor. All those green needles, they're going to be antimicrobial for a while. Once they start to break down and let some time go by, then the organisms are really going to take off fast. Yeah, alder is one of those great chips. I like using alder chips because they don't have a lot of antimicrobial compounds. They're one of those woods that you can use really, really fast. So I love alder chips. Alder chips are fantastic. And in fact, in California, where my wife and I come from, we didn't have access to alder. It was predominantly a lot of conifers, and eucalyptus was a massive amount that was there. There was a lot of, that's a year-long pile. It's going to quarantine there for quite some time before I can actually use it. It's a little bit multi-layered. But the question was this. I have comfrey. I now, there's a process where we can make a plant ferment where we can take comfrey, a little bit of woody material like dead leaves or wood chips, whatever else, and you put it into water and you let it sit for 3, 4, 5 weeks, whatever it's going to be, and it starts to have the smell to it. You're fermenting the comfrey. So can I use this as a plant fertilizer? In fact, that's exactly what it is. You're making a plant ferment that's going to solubilize the nutrients were in that comfrey into now a soluble form that could use as a plant fertilizer. So let me go back to this slide right here. All right. When we use compost that has organisms, what I'm trying to do is I'm trying to set the biology around the rhizosphere to have perpetual nutrient cycling, a resilient system that says, I've got the biology that can go mine the nutrients, I'm providing the food sources, and now I'm in a very resilient system, sustainable system that's going to continuously work. Plant ferments are a type of fertilizer. You as the person are now going to say, I think that plant needs the fertilizer that comes from that ferment, and I'm going to add it. And have you ever heard of KNF, Korean Natural Farming, JDAM? There's a bunch of these different types of fermenting processes. They're kind of cultural practices that use a lot of plant ferments. And I can tell you, I have some of my friends and some clients that use KNF, or Korean Natural Farming, plant ferments very effectively. Boy, their plants look good. They have intuitively figured out what that crop needs and how they make those plant ferments. But again, that plant is relying upon you as the farmer to make those ferments and to apply it when the plant actually needs it. And do you always get it right? Not always. In fact, sometimes you can add too much nutrient. There's this aspect of saying, well, if a plant doesn't have enough nutrients, obviously it's going to go under stress and it's going to be deficient. But if you add too much nutrient to a plant, it can block the absorption of other nutrients. So the plant may have a whole bunch of molybdenum, but you just blocked 2 other nutrients from really getting absorbed in that plant, causing a stressor to it. It's a very exactly. And your dilution, and the question was, should you dilute it, or it should be diluted for sure. But again, it's the principle of saying, you are the person that's now going to be responsible for making the ferment and feeding the plant when you think the plant needs the food, versus a system like this is saying, I'm just going to make sure the biology is present in the diversity and quantity and the ratios that allows the plant to control that nutrient cycling to occur. Different principles. I typically focus on doing this kind of work, adding extracts and getting biology, and not doing the plant ferments. I've actually made comfrey ferments and plant teas and so forth, and they've worked great for me. Well, yeah, there's this term that people will use around comfrey called dynamic accumulators. The idea is that they are really good at pulling nutrients from the soil and putting into the bodies of their leaves. There's been some debunking to that, saying that they don't really, accumulate nutrients any better than a lot of other plants that are out there. But what's special about comfrey is that it grows really fast. You can cut it 3 or 4 times a year and you can use it like crazy. And I'll tell you this, we have 5 fruit trees on our plot. We're creating an urban homestead in our new area that we have. And I have comfrey under each one of my fruit trees. When we lived in Auburn, California, we had 46 fruit trees and they all had comfrey underneath them. Comfrey is a fantastic plant. I just love, love, love comfrey, especially when planting with trees. They're a great understory. They grow really, really well. I can chop them. They suppress weeds. They're fantastic. Comfrey is a great plant. But don't, yeah. Well, yeah. So comfrey is a special plant too that the roots, if you break them up, they will propagate like right. If you're going to get comfrey, you try to get the Bocking 14 variety of it because it has less of that spreading capability. But yeah, comfrey is just like any other plant. People used to think we were crazy. We had our homestead and we grew a lot of produce on our property. And I loved using mint. Love mint. Mint is aggressive. It will take over. But in this food forest, I had mint and horsetail and all these other very invasive plants all competing with each other. And boy, I tell you what, that food forest was the worst plot of land on our property, and it turned into the most productive, and really in the 3-year period of time frame. We flipped the script, essentially. Sure, biodynamic. It's very different, although there's similarities in biodynamic. Biodynamic, with maybe not the intention that I use here, is still doing some very similar things. There's a little bit more aspects of biodynamic farming that I don't necessarily follow or use, but I have biodynamic farmers that do a fantastic job with managing, they make compost and they get good biology compost and so forth. So the question is, do I advise on breaking the soil or not breaking the soil? I am a very huge proponent of no-till. I do not like tilling. But I'll tell you this, almost, I'd say 80% of my clients, I've recommended to go in and till one last time. Compaction. There's a tool called a penetrometer. It's a ridiculously simple tool. It's just a tool, a rod that has a gauge on it, and you press it in the ground, and it tells you the resistance. How much compaction do you have? When I run into a lot of agriculture situations, the soil is compacted. And I could wait for biology to uncompact that soil for me, but boy, it can take a long time frame. What I will tend to recommend my clients do is to subsoil their soil. Break it up one last time, but I also say don't just stop there. When you're breaking that soil, we're going to add biology. We're going to drip an extract down into that broken zone, and then we're going to plant plants right away. Get plants in the system. That should be the last time that we have to disturb the soil in that way. Broad forking, if you could use that, again, broad forks is better than rototilling. If you take a rototiller, you're really messing up the soil. Broad forking is a little bit more fracturing the soil. But eventually, you should be at a spot where you don't have to worry about doing broad forking or anything because the biology is creating those aggregates in the soil. You could tell when you walk onto soil that's been very well inoculated with biology and it's created a lot of structure. It's very spongy. In fact, our garden beds, I don't use trowels. I just use my hands to plant things, it's because it's so friable and so loose that you can actually move it around. So the question was saying people have a very favorite place they like to forage and for mushrooms, and it has been recently thinned. I will give you my opinion. I don't know for sure, but the more trees that you take out of the equation, as long as it wasn't overly, if you have a lot of trees, sometimes the thinning can actually be a very positive thing, right? It can stimulate growth. But if you really thin it out excessively, then you may not have as many trees that are actually pushing out exudates to cause more fungi to grow. So it may lessen it. Right. Exactly. Yeah. So that's the kind of chicken or egg thing. If it was really dense trees and the trees were competing so heavily with each other that it was actually causing problems, then maybe opening up some of that forest land is going to cause the surrounding trees to really push out a lot more exudates, have more energy. It may be a benefit to that. There's a thing about setting things in balance and getting the ecosystem set the way it's supposed to be. In California in particular, it's a very fire ecology. If you were to go look at historical forests, the way that they were managed for thousands and tens of thousands of years was by using fire. Forests were very, very thin. You look at the forests now in California and they're very, very dense. That's actually one of the reasons why we have so many fires that are devastating in that environment, because it is a fire ecosystem, but now it's just so much fuel that's there. So I think there's a balance in there. Okay, Any other questions, or should we move to the microscope? If you guys don't mind, I'm going to pass around a few of these and just pass them around, and we'll take a look at them. And please do open them up and smell them. There is something about smell. You know, when I tell people to assess compost, to assess materials, you're going to use your eyes, you're going to use your hands, and you're going to use your nose. Your nose is a really key component to this. It should smell earthy. It should smell dank. It should smell, you smell it, it's hard to describe. You smell it and you go, oh yeah, that's the good stuff. When you smell something that's bad, you're like, that's not so good. Your nose will tell you quite a bit. Okay, so let's start with the depressing news first, and let's look at some agricultural soil. And in fact, you'll see it when it's coming around. You look at the color of it as well. So a couple things I want to point out to you. This is agricultural soil. It does not have a very little aggregation to it, mostly mineral, and it's going to be highly bacterial. So it's really even hard to see with this, but there is just a little tiny bit of bacteria floating around in here. As I'm just scrolling through here, I want you to look at it. No structure. You're probably not really seeing too much life in the soil. In fact, I'm seeing nothing but bacteria in here. And I'm sure it's not showing well for you guys yet because you haven't seen any of the life in the soil. But I just want you to kind of get used to the kind of structure of the way it looks. This is the compost I was telling you about. Wow. All right. Just from that view, you can see a lot more aggregation. See all the little darker areas that are up there? And in fact, we have an arcella right there. See that round circle? An arcella is a type of testate amoeba. Anybody remember what amoeba eat? They eat bacteria. Bacteria. So arcella, in fact, that organism right there, it has a big round test that we create. A test is simply think of it like a hermit crab. Everybody familiar with the hermit crab? Has a shell. A testate amoeba is the same thing. It's going to have this shell. Arcella being a very round, and it has an open hole. Right now, the hole is on the underside, so we can't see it. Bacteria just flows right into the arcella, and the arcella then just eats away. The arcella is the round ones. Now, see, right in the middle of the screen right now, do you see this other egg-shaped one? That right there is a testate amoeba, not arcella, but just a typical testate amoeba. And in fact, when I look at this, boy, it's still pretty, this right here is a testate amoeba. All of that is the amoeba itself. And right here is the opening. It will pull a plug of organic matter or aggregate, and it will plug up the hole. We shook that testate amoeba around, and it went, whoa, this is not hunky-dory for me. It's now in its test hiding away. If we let it sit here for a while, eventually it would push out that plug of organic matter, and these pseudopods would ooze on out, and it would start just then grabbing bacteria and pulling it into its body. Still feel like I could get this a little bit more focused.
And those underground things on the right-hand side.
Spores. Fungal spores. So, great question. The question was, what magnification am I at right now? 400 total X magnification. So I have a 40X objective and then 10X in the piece for the camera and also for my eyepiece. So it's 400X total magnification. So 400 times viewed in. That makes sense. All right, we're going to find some fungi though. Let's do that. Oh, there's some good fungal spores. Let's look at that. Right in the middle of the screen. Fungal spore. All these are fungal spores. And fungal spores come in all different shapes and sizes and colors too. In fact, it's great when you're doing spore prints. You can take a microscope and you can go really look at those spores. So you can see that type of mushroom has this type of looking spore. A lot of carbon material around the spore. And also then, boy, that's a good question. It's not a cytoplasm at that point. It's just genetic material that's when it wakes up, it will turn into the cytoplasm. That, you know what, I don't know exactly what the material is in there, but it becomes the cytoplasm that starts growing the fungal hyphae. Those are spores from fungus that have not germinated. And really, when I look at a lot of soils, in fact, this is my compost. there's tons and tons of fungal spores in here. They're just waiting for the right chemical signatures. A lot of the tiny little dots are going to be bacteria. So in fact, you can start, everybody can, if you, I use this trick, you defocus your eyes. So when you look at this thing, look at the vibration or the movement. Now, I'll tell you this as well, that small particles in water, there's a thing called the Browning effect. It simply is the vibration of water itself. So a lot of these could be bacteria that are not motile, they're just in there, but they're vibrating around. What becomes really difficult is when I look at like the agricultural soil, it's a lot of clay particles. Clay particles are roughly the same size as a lot of the bacteria. It's very hard to distinguish. I have to really dilute down those soils to like 1,000 to 1, 2,000 to 1. So I'm only seeing like 5 or 6 of those particles in a field of view. And then I'm looking, are they jagged? They're jagged, they're clay. If it's a cocci or a rod, it's a bacteria. All right, let's find some other cool stuff in here though. I'm going to get us back out here. I have nematodes. Oh, in fact, can you guys see that nematode right there? All right. Now we're talking. All right, so I told you there was 4 types of nematodes. One being root feeding, which this one is not because it came out of compost. They don't like to live in compost. But we have bacterial feeding nematodes, we have fungal feeding nematodes, and predatory nematodes. And the way we're going to find out what kind of nematode it is we're going to look at predominantly its mouth and its esophagus. And so let's zoom in a little bit here. So this right now is 100x magnification. So how's that coming across? Pretty good? Oh, I got to zoom in a little bit better. Let's see. All right, that I think is a little bit better.
Yeah.
All right, so take a look at this guy. Now I'm going to zoom in a little bit more. I'm going to go, yeah, bacterial feeders, they have a mouth. It's a very long, narrow mouth, and it's like they suck in the bacteria. They're sucking it through. A fungal feeding nematode does the same thing, but they use a stylus that is basically a spear that's going to puncture the fungal, the hyphae, and is going to suck out the cytoplasm. All right, so let's go in a little bit deeper in here. This is a good size. Oh, there's 2 nematodes. Oh boy, look at that. So remember that agricultural soil? Didn't look anything like this. What we really want to see is we want to see our soils have this kind of biology, the amoeba, the nematodes, and all of that. So let me go in now, really zoomed in.
Yeah.
All right. Yeah. So, and it's just going around and it's basically saying, you know what, I'm trying to find it. Oh, now let's see what it is here. Oh, this is a fungal feeding nematode. And if we get it just right, well, it's not piercing anything yet. There's a stylus that's right at the very tip of the mouth, and it looks like a little spear. You guys see it? It's hard sometimes to pick up.
So the nematodes actually look for their favorite food.
They do. They're going to go around and sense, and they're going to be, no, I don't want that one. I don't want that. Oh, there we go. Exactly. And, you know, fungal hyphae, very long networks, mostly empty. Well, it's not going to want to puncture an empty part of the fungal hyphae. It's looking for where the growing tip is at because that's what it wants. It wants the cytoplasm that's there. Chemicals. Yep. Mostly is just chemical sensing. They're a little bit, they're too small in that sense. But you know, their presence are going to cause bacteria to form aggregates that are going to, because they want the protection, which then fungi are going to wrap those aggregates into macro aggregates. And so they're going to help drive that biology to create that structure in the soil. All right, let's see if we can find some other cool things here. I want to find us some fungi.
So, nematode in the sponge.
Yeah. There's a type of nematode. It's a bacterial feeding nematode. It's called an EPN, an endopathogenic nematode. And what it does is it fosters a bacteria or bacterium that are really good at killing insect larvae and insect eggs. And what it will do is It goes up to an insect egg, let's say, deposits this bacteria. The bacteria goes crazy, consumes that egg, bacteria population explodes. It goes in and slurps up all that bacteria and says, thank you very much. I'll keep a little bit. Now we'll go to the next egg, and then we'll go to the next larvae or whatever it's going to be. We use the EPN nematodes all the time in agriculture. When you want to control certain pest populations, you time your introduction of those nematodes to when the flights of those organisms are there. All right. I don't want to disappoint you here, so I'm going to go to the forest floor stuff, and let's take a look at that. I'm going to show you guys some fungi. And notice there was fungi in there. You probably saw some of it. But boy, when we get to these, oh, what happened here? No, one of my slides got consumed by the other slide. So we'll take a look here. All right. So this came from a forest floor. In fact, I went to a park outside of Newport called Mike Miller Park. And I went ahead and got some samples. All right, so right there, right in the middle. Unfortunately, this projector is not doing a good job, but this is fungal hyphae right here. It is actually transparent. It looks kind of solid right now, and it has septi in it. But also, look, tested amoeba right there. So, even though the forest floor is fungally dominant, doesn't mean we don't have bacterial sources in the soil. It's still, the trees are still going to get some of its nutrient cycling from bacterial sources. So we should see amoeba and flagellates and those types of things. You know what? I'm going to go find us a really good section to go look at. All right, here's some good branching fungi. Oh yeah, we got some good stuff up there. All right, let's go take a look at this. Wait a second, where is that at? There's some right there. Okay, so you guys can see that branching one right there. Now, another interesting thing about fungi, so there's a lot of material that looks like fungi in the soil, and that's just organic matter. It could be cellulose, it could be whatever it's breaking down. But in particular, fungi, when they break, they usually cut clean edges. And so a lot of organic matter, when it breaks, it shreds at the ends. And so sometimes I'll be looking at, especially like a colorless fungi, I'm like, is that organic matter or is that a fungi? I'm always looking to see what kind of clean break that it actually has. And so here's another fungi. You guys can see that one? I have some better sources here though. I'm going to do this. I'm going to go there first. There we go. All right, so you can see that really big, large fungi right there, right? Let's go in and take a look here. It's a little too dark for you guys. You can't see the septi, right? Yeah, it's a little too dark for that. All right, what about here? Can you see the septi in that one? Oh, bummer. See if I can find another one oh, now, see this right here? So this is a weft of a bunch of the same type of fungi. If I look at this, it's probably from a singular or the same species of fungi because it's all the same color, same diameter. And you can just see that sometimes you'll get in these kind of wefts. It could be also the start of forming of mycelium, right? So you see all of them combining together. In fact, when I look at mycelium, when it starts to form like this, it has that kind of structure or shape to it. Sure, an aggregate, or you mean like a mineral? Yeah, they're shiny, usually very, very shiny. In fact, right there's definitely some minerals. That's a mineral particle. That's a mineral particle. A bunch of these are mineral particles that are in there as well. Unfortunately, the projector is not doing a good job of showing the crispness of it. If you look through here, you would definitely see it. Yeah. Oh, there are mineralogists that for sure that's exactly what they do. I cannot. Yeah, it's just like fungal spores. There are mycologists that are really good at identifying fungi through fungal spores. Really hard to identify fungi by their hyphal structure. because a lot of fungi will have very similar in the hyphal structure. There are some exceptions to this. Like if I see a fungi that has what we call a clamp connection, which is basically fungal hyphae with a bubble around the septi, basidiomycetes. Now, do only basidiomycetes make clamp connections? No, but they are a type of fungus that definitely does make clamp connections, and the others that do are much more rare or unique and that type of thing. So if I see clamp connections, I'm almost, well, that's probably a basidiomycetes that I'm looking at. All right, let me go back out here. Let's see if I can find another interesting thing of fungal hyphae. I want to find a good light colored fungal hyphae that you can see the septi in, maybe right here.
Slide at the
Oh, when we were just looking at it? Oh,
color.
here we go.
It's not so good.
I don't see any really uniform septi. This could be from a mycorrhizae fungi.
That wasn't the one, sorry. You'll
No,
find it,
right.
but saw it.
Oh, can you guys see that little thing moving around? It's let me get focused on there. See that thing just bumbling around a little bit? No, that is a flagellate. Bumbles. Yeah, question?
Could that be a slime mold?
Right here that was moving around. No, that's a flagellate. And the reason I know it's a flagellate is you can't see it on there, but in here I can see the flagellum. And so the flagellates will usually have one or two flagellum, one just on the back, or if they have two, they'll usually have one in the front, one in the back, and they just bumble everywhere. Sure, they're very large though, really, really large. Yeah, I would have to be at like the 4x magnification. The microarthropods that you can, or the arthropods you can see with your eyes are huge when you look at it here. Yeah.
So, yeah, slime mold would be flagellant? Sometimes
I'm trying to remember on slime molds. I think they,
Sometimes not.
right, they do, right. And I'm trying to remember the motility. I remember looking at it a long time ago, a slime mold. You piqued my curiosity. Now I have to pull some slime mold in there. If I do, I'll give you an email and send you pictures. Yeah, exactly. So I'm trying to find some other, and now one thing I want you to notice, like in this screen here, there is a lot of different type of fungi. There are different colors. There's a clear one towards the bottom. You can see the fungal hyphae right there. There's more colored ones right there, darker in color. This is common when I look at forest floors. Just a massive diversity of different types of fungi. When I go into a lot of agricultural situations, diversity is just not there. What are you looking for, sweetheart?
She's looking for some that.
Yeah, the projector's making it really, really hard to see. Let me see if I can. You might be able to see it on this one right here. There's a septi right there.
Yeah, there's one.
Right, and that's just that cross wall. If we have time, oh, there's one right here. So you can see this. See that right there? That's the septi. That's that cross wall that you can see. They just vary. And a lot of really beneficial saprophytic fungi have very uniform distances in their septi. When you get to a lot of the disease-causing fungi, they usually won't have a color to them, and they won't be uniform in diameter, and they'll also not be uniform in septi or have no septi at all. Clear, exactly. Now, if I see a fungi that's clear, not uniform, doesn't have septi, does it mean it's disease-causing? No, but chances are it's going to be in that disease-causing kind of classification.
Disease-causing ones are kind of herky because they're opportunistic. And so they're growing quickly, they're slowly.
I think that's part of the equation. The other part of the equation, disease-causing fungi tend to thrive in anaerobic soils. Their structure tends to be different. What they're receiving and how they're actually performing and growing is going to be from different conditions than like a very aerobic condition. Typically, when the soil is very aerobic, disease-causing fungi won't grow. When it becomes very compacted, compaction equates typically to anaerobicness because compacted soils don't have structure to them. They don't allow for the oxygen inflow. So you see different types of organisms in anaerobic soils than you do in aerobic soils. So when I'm looking here and I start seeing a lot of the disease-causing fungi, the clear, colorless, not uniform, and then I start seeing lots of ciliates. These are types of protozoa that thrive in anaerobic conditions. I start seeing those conditions, I'm already making some assumptions of saying, I think the soil is probably anaerobic. And more likely, that it's anaerobic because of compaction. And so now we're saying, well, how do we resolve the compaction issues? The subsoiling I talked about, or we're just over time, we're going to try to keep on getting the biology to do that work for us. Okay, let's see, what else do I want to show you here? I know we're kind of getting close to the end here. Oh, there's the nematode, but he's just going crazy in there. So more than likely, if I was to look at this, it'd probably be a fungal feeding nematode, but it's kind of the edge of the slide. Do you guys see it? Yeah, and this is a good sign to see. I mean, when I go look at really healthy forest floors, you see that constituent population of nematodes and protozoa and different types of bacteria and fungi and so forth. But if nobody told me where the soil came from and I looked at it, I would say, oh, this is probably pretty darn healthy soil. This is probably something from a healthy ecosystem.
Why? Is it just small one?
Oh, the one that's wiggling around, or you talk about the one that's like below it?
One that just, it's very, it's coming back and forth. The one that's.
Yeah, that's a nematode. And let me see if I can. Oh, nematodes come in all different shapes and sizes. This is a relatively large nematode. Look at the tail. More than likely, but I've seen bacterial feeding nematodes. Sometimes the predator nematodes are huge, almost double the size. You can find nematodes that are twice that size. But then I also will find fungal feeding nematodes that will be a quarter of that size. So there's many different species of nematodes. That's aggregates. Yep, exactly. We're forming aggregation in here.
Do you find microplastics in soil often?
Yes, and it's unfortunate. You can usually tell them, but pretty easy to see. The question was, can you find microplastics in soil? For sure. It's there. They're very odd shaped and they're colors that just aren't natural. You're like, okay, this does not equate. Yeah, oh yeah, for sure. Yeah. Well, fortunately, on the projector, you guys aren't seeing color. If you're looking through here, you'd be like, wow, there's all kinds of colors in here. So yeah, exactly. Well, you know what? There is a lot of work right now. So a lot of the work that I do is I will make very precise dilutions of materials, and then I will put a single drop on a slide, and then I will look at this right now is what we call a field of view. That's that nematode. Oh, right there. No, right there. That right there is a big flagellate. Yeah, its flagellum is big. It's right in front of it. And you see the fungal hyphae, you can see the septi in this one, right? Can you see the septi? Kind of. Let's see. There we go. So here, fungal hyphae, septi, 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 right?
There's just little dashes.
That's another flagellate that's moving around right there.
And they usually are all even. You know.
there's
Looking at it, not 6 inches apart, but just like each of them are 6 inches apart.
Another flagellate cruise around. Yeah, they bumble. They're easy to identify a lot of times because they're very unique in their motility. They bumble around in the soil very, very easily. What was the question?
Equally apart from each other.
Right.
Is that structural?
It's structural. The septi are meant for structure, to maintain the structure of the fungal hyphae. And like I was telling you about basidiomycetes, how they create a clamp connection. Unfortunately, I haven't seen one yet, but what you'll see is right where that septi is at, a bubble. And what happens is a lot of times when the cytoplasm, when it's done growing, let's say right here, the cytoplasm said, you know what, I don't like this environment anymore. I want to go back and go to another location. The cytoplasm has to travel back through, and it has to travel through those cross walls. A lot of times the cross walls will have some amount of permeability to it and they can go through. Basidiomycetes, the cytoplasm is really big. it can't fit through these cross walls. So it creates a clamp connection, which means it goes right through the clamp and then over, right through the clamp and then over. And then whenever we see branching, that's because it basically said, you know what, I'm going to now grow this direction, and it'll start putting on fungal hyphae in that direction. Okay. Well, I know we were probably getting close on time, right? So any other questions? Does anybody want, before I close the microscope, if you want to take a look through? Oh, for sure. Oh, yeah. You want to look at that one again? I will put that one up. But I will, no, that's fine. What it looks like, yeah. Unfortunately, there's no sniff-o-vision, so you can't. That's, yeah, this is what I'm going to put up right on the slide again. So let me go back here. And I'm going to find us something interesting. So here's a nematode. Here's a nematode that's sleeping right now. Yep. Yep.
Do you find that the temperature of the lamp ends up killing off specimens?
No. I use LEDs. So these are LED lamps. The question was, does the temperature of the lamp on my microscope have any kind of impact to the organisms? No, I use an LED. Even when I had a bulb, it wasn't strong enough to be able to do that.
Okay.
Yeah. So when we're looking here at a nematode, unfortunately, you can't see the mouth. You can't see the mouth of this one, but I know it's a bacterial feeding nematode because right here is an esophagus. And it's called a, well, this is the esophagus right through here. This is the terminal end of the esophagus, and it has what they call is a cuticular valve. And what it does is it sucks in the bacteria. When it gets to this spot right here, it crushes. And you can see it when it's actually working on bacteria, it's pumping, and it's crushing bacteria before it goes into its, exactly. Yeah. So not only the mouth part right now is hidden. I can't see the mouth part, but I know that only bacterial feeding nematodes have cuticular valves. And you can see there's a little T right there. That's where that valve is at.
So what's the rest of it? Just
Digestive. And then sexual reproduction organs. Sometimes you'll see eggs in the nematodes. Yeah. Sometimes you'll see the nematode eggs with little nematodes inside of them that are getting ready to hatch. There are very cool organisms in here. Everybody heard of tardigrades? Water bears? Oh, they're so much fun. Some of the best places to go look at organisms in a microscope is go to a wetland. Oh, go to a wetland and there are all kinds of things in there. There's algae and there's rotifers and ciliates galore, all kinds of stuff.
But I've never seen them in
Yeah.
Any final questions?
Any final questions? Thank you. Appreciate it. Yeah, thank you. Thank you guys for having me here. Gracious host, and I'd love to be able to share knowledge.